Hi, and welcome to this mind map where we're going to be going through Module 6.1, Cellular Control of the OCR A-Level Biology syllabus. So first thing is, before you do this, you might want to consider going over the nucleotides mind map as well, because it really ties into this module. So first of all, make sure that your knowledge of DNA is really solid, and then we should get into this mind map. So if you're ready, let's get going. Oh, almost forgot to say that the link to the blank mind map you will find in the description of my video, and I'll also paste the link underneath this mind map. So if you go to that, I think it's the last slide in the slides deck, you can print this out in color in A3 would be best, and then you can annotate all around it. Okay, let's get going. So let's start with a quick recap on DNA. Here we have DNA. And of course, this stands for deoxyribonucleic acid. I'll write that up here. So deoxyribose is the sugar, and it's a nucleic acid. So if we zoom in and look at the molecule in a bit more detail, we've got the sugar phosphate backbone. Let's label it in a different color now. Over here, sugar phosphate backbone. We've got the kind of bases, and sometimes they're called the rungs of the ladder. And then if we look here, we can see which bases match up with each other. So A to T always match. So let's kind of label that over here. This is kind of recap. A to T, and there's two bonds there, two hydrogen bonds between them. And C to G always match, and there's three hydrogen bonds there. Two H bonds, three H bonds. And this is more of a recap for the last module, but A and G are the purines, and C plus T are the pyrimidines, and you always have a purine with a pyrimidine. And that is so that the rungs of the ladder are the same length. So the purines are the slightly larger ones. You can see that G is a double ring structure here, as is A. A is another double ring structure here. So let's just kind of do that there to signify that. That little bracket around there, and on C and T I'll put a blue box around that to signify that those are single ring structures there. The sugar, as I said here, is deoxyribose. And we also have a phosphate group. And the DNA backbone runs 3' to 5' in one direction, and 5' to 3' in the other direction. So we call this whole thing an anti-parallel orientation. It's an anti-parallel and it's a double helix. As the ladder, which is formed by the phosphate backbone kind of sides of the ladder, and the base rungs of the ladder is twisted to form a double helix. Okay, so that's DNA structure, and perhaps you knew that already. But the first part of this module 6.1 is about mutations in DNA. So let's zoom out. And specifically, we're going to be talking about gene mutations. So that we can kind of branch out from here. So DNA, gene mutations. A gene mutation is a change in the DNA. Change in DNA code. And what causes them? So let's go below. Causes. Well, first of all, some of them occur spontaneously. We can pinch in a little bit more. During replication. So there is an error rate. So when DNA polymerase copies DNA, there is just an error rate is pretty low, but sometimes the wrong bases are put in, and that causes mutation. And that can only be inherited if it occurs in what's called a germ cell, so a cell that's going to go on to become a sperm cell or an egg cell, and that can then be passed on. But it could be a mutation in a body cell, and that might give rise to cancer, for example. So there is a spontaneous rate of mutation, and there are also some things that increase this rate. So these are called mutagenic agents. Will increase the error rate, and those things are, for example, UV light, radiation, and some chemicals. Now, you might have heard of the word carcinogenic agents, and carcinogenic agents is a subcategory of mutagenic agents that specifically cause mutations in genes to do with the cell cycle, and they can cause cancer. So I'll put kind of maybe brackets under their kind of carcinogens, or a subfamily. Okay, well, mutations may be a big deal, but they may not always be a big deal. So to understand why, we have to think about the DNA code. Coding for information, we'll call it. So up here, coding for information. And the DNA code is a couple things. It is, number one, universal. So that means that all living things use exactly the same DNA code. So if there are 3 bases in a sequence, for example, TTC, that would, in every single living thing, that would code for the same amino acid. So if I pinch in here, this is a little bit blurry here, but TTC, first position T, second position T, and the last position C, that specifies this amino acid, and I believe that is phenylalanine, although it's a bit blurry, but that would code for phenylalanine. That TTC sequence of bases would code for phenylalanine, everything from a plant to a bacteria to a mammal to everything. Okay, so it's universal. Second thing is it is degenerate. So that needs a bit more explanation. So that means multiple codons equal the same amino acid. Alpha alpha is a shorthand for amino acid, the same amino acid. So for example, if we pinch in again, sorry for the blurriness, if we look at this box here, there are actually, oh my gosh, there's actually 6 different codons for the amino acid leucine, which I think is the most, yeah. So like, for example, if you had T, T, and then A, that would be leucine, that's this one. Or if you had C, T, and G, that would also be leucine, that's this one here. So there are multiple codons for the same amino acid, sometimes as many as 4 or even 6 different codons mean the same amino acid. And then the last thing we should say about the code is it is a triplet code. So that means 3 bases is called a triplet and codes for the presence of 1 amino acid in a polypeptide. Okay, so if we have a gene mutation, pinching out a bit, which is a change in the code, then there are several different ways that this can play out. So we're going to look over here and look at different examples of gene mutation. So the most common type of mutation is called a point mutation. And this is just a change in 1 letter. One base, let's say. So what happens in a point of mutation depends specifically on the letter change. So over here, This is called a missense mutation. Missense mutation. And a missense mutation basically means that we change an amino acid. So here, in the original sequence, the codon reads TTA, and I'll write that above in case it's blurry. And in the new mutated sequence, it's TCA. Now that changes an amino acid. Changes the amino acid here. This says leucine, and this one says serine. So we have changed amino acid. Now this could affect the protein structure. It doesn't have to. The tertiary structure. It will definitely affect the primary structure. So it changes the amino acid, changes the primary structure, and then that may mean that the protein doesn't fold correctly, which may mean that the tertiary structure is messed up, and the protein doesn't work. So if you get an exam question on this, you say the amino acid is, well, the triplet is changed. This means a different codon and a different amino acid is inserted by the ribosome in the polypeptide chain. This means that the protein's primary structure, secondary structure, and tertiary structure are all changed. There may be a mark for each one of those things. And then the protein may, if it's an enzyme, it may not be the right shape. It may have the active site may be the wrong shape for it to work. Or if it's another protein, it just may not be the wrong shape for it to kind of fit wherever it needs to fit. So that's a missense mutation. So that potentially could be quite bad. Over here, we could have a silent mutation. So silent mutation is one that it's kind of the body almost doesn't notice it. So a silent mutation, there is no change to the amino acid. And that's because even though we have a change in the triplet sequence, or the DNA sequence, here it's TTA, here it's TTG, the TTG it codes for the same thing as TTA. So TTA is the leucine, that one there, and TTG is also leucine. So the same, so we'll just label that here. Different triplet, oops, same amino acid. And the word that you need to use when describing silent mutations is degenerate. It is because the code is degenerate that you can get silent mutations. So silent mutations, basically the body probably wouldn't know, there'd be no consequences for the cell. Okay, now we have some other types of, there's one more type of point mutation, which is called a nonsense mutation. Down here. And this is actually potentially quite serious. Well, they're all serious, but this is a serious one. Because it just cuts short the protein. We call it a truncated protein. And that is basically too short. It has been cut. You're supposed to have a protein is like a piece of string that's all coiled up, but when that string is being sort of wound, it gets cut short. The string is not the right length. So when it coils up, there's a part missing. And why that happens is because we change to a stop codon. So codon changes to stop. And if we have a stop codon, what that means is the ribosome stops adding amino acids when it gets to that stop. So the ribosome stops building the protein. We could write underneath. Ribosome stops adding amino acids. So there are, it stops here, this one are all missing, those amino acids. So we could have a protein produced that is, many hundreds of amino acids too short. So it's obviously not going to work properly. And then the final thing is called an indel mutation, which results in a frame shift. So we can either plus or minus base added. So what I'm saying there is that we've either added in an extra base or deleted a base. So for example, up here it should see TTA CGC, but here it just says TAC GC. So basically this T, I'll just write here, one letter T is deleted. Now, because that one letter T is deleted, when the ribosome is reading the code of the mRNA, it doesn't kind of know that a T is missing. So it kind of reads the 3 letters shifted 1 letter back or forwards. So every single codon from that deletion onwards is the wrong thing. So this again is very bad for the protein. Like the whole structure of the protein is going to be all messed up because look, this amino acid is different, this amino acid is different, this amino acid is different. So we could maybe add down here every amino acid. And sometimes biologists use a word downstream. So I would say every amino acid downstream, which means along the code, is affected. And that would be if it was an insertion or a deletion. Okay, so those are point mutations. But I wanted to spend a little time as well on other types of mutation which can occur. And the first one is a triplet, triplet nucleotide repeat. Now, in some genes, there are naturally occurring repeated sequences of letters. So I can't remember, this is too blurry to see, but let's just use the example CAG. CAG is the most common repeat. So in some genes, they have just CAG, 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 repeated many times. So this is repeated many times in a normal protein. One example of a protein with this in is there's a protein called Huntington. And this protein is the one that is mutated in Huntington's disease. So in Huntington, there are normally between, I think, 6 to 35 copies of this repeat, of the CAG repeat. But when the protein is, when DNA is copied, sometimes the number of repeats is increased. Okay, this is quite an easy thing for the DNA polymerase enzyme to do, basically. And if it's increased too many times, then the protein becomes too large, can't fold properly, and then that's sort of mutated Huntington. So mutated Huntington can't fold properly. And forms clumps of protein, kind of plaque, which kind of destroy or cause damage to brain tissue. And this causes like neurodegeneration, like your brain kind of breaking down, and eventually leads to death. Now, how many copies of CAG do you need? Well, it's between, I think, 40 to 60-ish copies. And there is some sort of general, it's more than 60, 40 up to 120. And basically, the more copies of that CAG repeat, typically speaking, that means the earlier people get Huntington's and the more severely it progresses. So sometimes people get like 39 repeats and they may or may not develop Huntington's. So Huntington's, remember, coming up in your next module, 6.2, Huntington's is a dominant inherited genetic disease. So there's one example of how triplet nucleotide repeats gene mutation and how that plays out. The final thing, which actually I realise now is not in this module, it's actually 6.2, but I'm talking about mutations, so I feel like I should put it in here anyway, is a chromosome mutation. More on this in the next module. So this is when we're talking about generally things that go wrong during meiosis. Problems in meiosis lead to too many chromosomes being inherited. So one word related to this is polyploidy. And this is when an organism starts out diploid, but due to a severe mess up, meiosis, basically, like you have diploid gametes joining together. So this is like a diploid sperm. It normally happens in plants, but let's say sperm anyway. Moss actually do have sperm, so let's say this is moss. Moss sperm and moss egg. So this is the male gamete here, and this is the female gamete here. And they're both diploid, and then they come together and they produce a 4N new organism. And in mammals, that would just kill the organism. There would no way it would survive. But sometimes in plants, you get tetraploid plants that do actually exist. And actually, breeders of plants sometimes use this to produce new plant varieties. So sometimes you get like orchid breeders like causing this to happen to get new fancy looking orchids, basically. Much more common in animals is this over here, which is called aneuploidy. So polyploidy, let's just define that. It is an extra whole set of chromosomes. Like 3N triploid or 4N tetraploid. Whereas aneuploidy, which is this one over here, is not an extra whole set, it's an extra chromosome. So we're not triploid or tetraploid, it's just that there's 3 of one chromosome is the most typical. An extra single chromosome. And the most common one here, you can just about see, is chromosome 21, e.g. 3 times chromosome 21 equals Downs. So Downs is caused normally when, well, is always caused when an egg, female gamete, contains 2 copies of chromosome 21 by mistake. Remember, it should only contain 1 copy because it's a gamete, so gametes normally have 1 of each chromosome. But what happens is there's something called a non-disjunction at meiosis. So that means at meiosis 1, when you've got the bivalent 2 chromosomes together, maternal and paternal chromosome together, they're supposed to separate at metaphase 1, but they don't. They get stuck together. They both go one direction. So then the gamete that's produced, the egg that's produced, has 2 copies of chromosome 21, and then the sperm cell brings another one, giving you 3 copies of chromosome 21. And the embryo will survive, but there are various health complications caused from this. Okay, so people with Down syndrome can live really, really full and happy and successful lives, but there are health problems associated with it, such as early death, heart failure, and other effects. Okay, so before we move on, it's important to say that not all gene mutations are harmful. So first of all, some gene mutations can be like not a big deal. Basically, they have no ill effect. So for example, humans, we're mammals, we're descended from other mammals, like, for example, rodents. Sorry to break it to you. And rodents can have like, I can't remember how many genes, but thousands of genes, let's say 5,000 genes associated with smell. They can smell a ton of stuff. Now humans in our evolution, a lot of those genes that were responsible for being able to smell all sorts of weird things have been mutated and they no longer work. They are now called, they're kind of like dead genes. We've still got them in our DNA. They're called pseudogenes. They don't work, they're not functional. But the thing is we don't need them because we have, better brain power and we have better vision. That can mean that we can look at a fruit to tell if it's ripe as opposed to smell a fruit to tell if it's ripe, for example. So, a gene might be mutated, but it might have no ill effect because we don't really need it anymore. In some cases, a mutation is beneficial. So evolution works by mutation. A mutation in a protein might make it slightly better at doing something, and that might be a beneficial mutation. Okay, so what is this central image in the mind map? This is the kind of story of how gene expression is controlled. Okay, so genes are in the nucleus, but are they actually expressed? Are they actually turned into a protein? Sometimes yes, sometimes no. What controls that? How do we stop a gene from being turned into a protein, or kind of start a gene being turned into a protein? Because remember, every single cell in the human body has got the same DNA, more or less. So I have around about 75 trillion cells in my body, and each of those cells contains roughly the same 25,000 genes, more or less. Okay, but my cells in my brain will be using different proteins than cells in my liver, and they'll be using different proteins than cells in my muscles. So we need to be able to turn on and turn off different genes in different places. And there's a bunch of different ways that we can do that that you need to know about for this module. So before we actually talk about human control of gene expression, we're going to look at a case study of bacteria. There is a human parallel. So this box here is going to be control of transcription, let's call it. So whether or not we decide to turn the DNA code into messenger RNA. And there is a sort of, before we look at how that's done in mammals and humans, we're going to look at a bacterial case study. And it's called the Lac Operon. Okay, so we're first going to look at a quick animation of the Lac Operon, and then we're going to label this and sort of talk about it. Okay, so first of all, what is the Lac Operon about? The Lac Operon is about a bacteria not wanting to produce loads of proteins if it doesn't need to. E. coli is a very common bacteria. Now, normally it will be respiring and breaking down glucose, and it produces certain enzymes pretty much all the time to break down glucose. Now, it can also break down and metabolize lactose, but it won't bother producing the enzymes to do so if there's no lactose around, because lactose is kind of like a rarer molecule. So it will only start producing enzymes to break down lactose if it detects a small amount of lactose present. That's good because it means it doesn't waste energy producing enzymes it doesn't need to. So how does this work? So the bacteria, in order to break down lactose, needs to produce two different proteins. First of all, it needs to produce a protein called lactose permease, which is a membrane transport protein, and it helps transport lactose into the cell. It's a carrier protein. It also needs to produce an enzyme, beta-galactosidase, which is an enzyme that will break down lactose, which is a disaccharide, into the two monomers, glucose and galactose. So let's watch how it does this. Now, the genes that code for all these proteins are kind of right next to each other in the bacterial circular loop of DNA. So we call these genes an operon because they're kind of next to each other and they operate together, basically. That's what we call an operon. So in this operon, we have a few different regions. We have P, the promoter. We have O, an operator region. Now, these P and O regions are not, they don't code for proteins. They're involved in the switching on and off of the operon. Then we have Z and Y, which are called structural genes, because those are the genes that actually will get turned into proteins, the lactose permease and the beta-galactosidase enzymes. So how does this work? We also have a regulatory gene, which is not next to the operon, but a little bit further away. So under normal circumstances, the regulatory gene is always transcribed and translated to produce a protein. So I is the gene, and it codes for the protein. So the repressor protein is produced. Now this repressor protein, under normal circumstances, will bind to the DNA. It has a DNA binding region in it, and it actually binds onto the DNA right here, kind of overlapping it binds mainly to the operator region, but it also kind of overlaps and covers the promoter region over here. So what does that mean? Well, that means that if there's no lactose present, in inhibitor protein blocks RNA polymerase from accessing the promoter. So RNA polymerase is the enzyme that would transcribe the genes Z and Y into messenger RNA, but it can't bind to its promoter because this is blocking it. So under normal circumstances, the repressor protein binds over PNO, which means that RNA polymerase cannot bind, does not transcribe the structural genes, so no proteins produced from Z and Y. This saves resources. Now, if lactose is present, something changes. So if a little bit of lactose is detected, tiny bit of lactose will just about seep into the bacteria at very low levels, even if these proteins aren't present. What then happens is lactose actually binds to the repressor protein, and the repressor then changes shape slightly. I'll show that again, and then it unbinds the DNA. So now the promoter and operator region are kind of free. So this means that the RNA polymerase can bind to PNO, and we get the transcription and translation of the structural genes, which means that lactose is taken into the cell and then it's kind of broken down. So let's label this. Perhaps you've already done this. So I is the inhibitor. And I'll put over there codes for repressor. P is the promoter. O is operator. Z is one of the Z and Y are the structural genes. So I'll just put it like that. I always forget which is which, but I've labeled it down here. So Lac Z, when everything is kind of working and producing proteins, Lac Z is going to be the beta-galactosidase. And Lac Y is going to be the lactose permease protein when they are transcribed and translated. So under normal circumstances, here we have a ribosome. Here we have transcription. This is messenger RNA from the inhibitor region. The ribosome produces this repressor protein, which binds and blocks the promoter. But if lactose is present, so this little red molecule here is lactose. If lactose is present, and here's again lactose is binding, that means that the repressor protein can no longer bind here. So then P here, this would be the RNA polymerase, is this molecule right here, this little yellow thing. I'll just highlight that. So RNA polymerase can bind, which means that when it binds, it's going to bind and transcribe all this to produce messenger RNA. And actually here, this messenger RNA you can see as a mRNA of 2 genes together. And sometimes bacteria do that. And a ribosome will then take that long messenger RNA and produce 2 separate proteins from it, one of which is beta-galactosidase. I'll just put BGAL for short. I've written it up here in full. And this is Lac Perm for short. Function of that, this is enzyme, and it takes lactose and converts it into beta-galactose plus glucose. You do need to know that. That's on the molecules unit. Is covered in the molecules Mindmap. You need to know the different disaccharides and what they're composed of. And then lactopermease is a carrier protein. Okay, so that is an example of how transcription can be turned on and turned off like a switch, basically, in response to the presence of lactose. If lactose is there, then the genes will be transcribed and the bacteria can use lactose as a respiratory substrate. If lactose isn't there, it doesn't bother wasting the energy. Okay, so this is a good thing for the E. coli bacteria. Okay, so that's Lac Operon as a case study of how we can switch on and switch off genes in a prokaryote. But why don't we talk about this in a eukaryote? Why don't we talk about this in a human? Well, basically, humans are way more complicated. So control of transcription works in a similar way to this in humans, but it's more complex, and it's kind of too complex for A-level, to be honest with you. So in eukaryotes, the first three, why is it different? And one of the main reasons is it happens in the nucleus. So in bacteria, remember, there is no nucleus. So everything in the cell is floating about and can sort of bump into each other. There's no separate compartment for the DNA. So the fact that there is a nucleus makes things a little bit different in eukaryotes, and the way that DNA is packaged and things like that makes it a bit different. There's also different enzymes involved. So in eukaryotes, the common feature is that transcription is turned on/off by transcription factors. Let's highlight that word, transcription factors. So transcription factors are DNA binding proteins. So they can bind to promoters to allow the proteins, to allow the DNA to be transcribed into messenger RNA, or they could bind to promoters to kind of block it from being transcribed. So DNA binding proteins and transcription factors, I'm reading here, are around about 8% of human genes, which is loads. So you can kind of get an idea that really the reason that we're so complicated and sort of amazing creatures is that it's kind of to do with our ability to regulate genes. When people first sequenced the human genome, they thought there would be loads more genes than they were. Some estimates were like humans must be at least 100,000 genes. And I think the first estimate was like 30,000, and then it's come down a little bit since then. And people were kind of amazed that we can be built with so few genes. Well, the reason is we're also really good at regulating the switching on and off of genes. So transcription factors are part of what makes humans so complicated and so amazing. So let's look at a animation now. Here's a video of transcription, okay? So this is just the transcription of any gene, but this specific gene happens to be a gene involved in bone strength. And this gene is actually sort of turned on partially by vitamin D. So if vitamin D is present, the gene is activated. We can see these proteins kind of binding to the DNA. Now these are all transcription factors, and they all have to assemble to start transcription. I'm going to show you that a little bit again, just to back it up a second or two. So the gene will only be turned on if the right number of proteins come together and trigger the start of transcription. And once transcription is initiated, off the RNA polymerase goes, and we're getting the gene transcription. So all those things before were transcription factors. Now here's transcription. This is RNA polymerase. So we can see DNA entering from the right and RNA, messenger RNA, exiting above. It's really, really fast. This is actual kind of real life speed. And let's slow it down a little bit and kind of look at what's happening. So inside that RNA polymerase molecule, free RNA nucleotides fly in or float in, and they pair up with the opposite base. So A's bind with T's and C's bind with G's. If we speed it up, we can see this is happening incredibly fast. So this is RNA polymerase. and the messenger RNA is exiting out the top. That messenger RNA formed in the nucleus would have to leave through the nuclear pore. So transcription can only work if those transcription factors and if the RNA polymerase can actually get to the DNA, to the gene that needs to be transcribed. So before we can transcribe a gene, the chromatin where the DNA is packed has to be in the right conformation. So now we're going to look at DNA packaging. DNA packaging, it's packaged as chromatin. So I'm going to go above. Chromatin. Chromatin is DNA. I'm going to zoom in. Chromatin is DNA plus histones. Histones are proteins. So DNA is tightly wrapped around histone proteins. And if it's tightly packaged here, then there's no transcription. Because basically, no enzymes involved in transcription, RNA polymerase, for example, they can't actually access the DNA. And it's only if it's kind of loosely packaged that we can get transcription. So controlling how tightly DNA is packaged is one way that we can either allow genes to be transcribed or expressed or block their sort of expression. Let's look at that animation now. Okay, so here we are. Here's a computer simulation of DNA. I'm just going to talk you through it. So each of your cells has almost 2 meters of DNA, and it would never fit unless it was packaged. So how is it packaged? Here we can just see the double helix, and there, just clamping down on top, is one of these histone proteins. Now the histone proteins kind of clump together and cause the DNA to wind around it, and they actually kind of wind around these histone kind of blobs, and they kind of form this kind of beads in a string configuration. And then these beads on a string coil up into this kind of quite fat coiled molecule. Now you can sort of see flapping about in, to either side of the DNA molecule, these little wiggly blue tails. I'm going to take it back just a second there. I'll take it back to almost here. Okay, you see these wiggly blue tails. Okay, these are the histone tails. And by adding chemical groups to the histone proteins, either adding groups or removing groups, the cell can kind of regulate how tightly packaged the DNA is. So here we're seeing the DNA package up really tightly, winding, coiling, and then super coiling again. And this actually is only happening when we're actually coiling up into a chromosome so that the DNA can be moved about during mitosis or meiosis. So typically speaking, the DNA would not coil up so much to be a chromosome like this, but would be more loosely packaged. Okay, so let's label these diagrams up here. So DNA packaging, or the science of the study of this and how this kind of can control access to the DNA, is called epigenetics. So epigenetics is kind of off syllabus, but in many textbooks it's mentioned as sort of a frontier of research, and it is possible that you could get some questions on epigenetics as kind of an application question if they kind of gave you a bit of information and asked you to kind of apply your understanding to it. So it's worth knowing about. It's also something that could come up at interview if you're thinking about doing biology or certainly biochemistry at university. So epigenetics is how genes can be switched on or switched off sort of somewhat permanently, semi-permanently. This switching on or switching off of genes can control differentiation. So let's just call epigenetics is switching genes on, off in a semi-permanent fashion. So this can control differentiation of cells. So this is why you can't necessarily get a skin cell to become a brain cell, because some genes have been switched off or switched on sort of semi-permanently, and it's very difficult to kind of undo those switches. And there are some studies that show that epigenetic switch on or offing of genes can actually be passed down 1 or even 2 generations. So some environmental exposure that maybe your grandmother or grandfather was exposed to, potentially stress, can cause changes in gene expression in you. Okay, so your grandmother or grandfather's experiences may somehow affect your genes, the way they're expressed. Okay, so this is sort of contentious research. Go and look into it. I'm not up to date, but you should explore it. So how does this work? Just quick little things about it. One thing, it can work by DNA methylation. So cytosine C can be methylated, which means there's a little methyl group added to it. There it is, just a little methyl group. And that methyl group there packs the DNA tightly. So therefore, if genes are methylated, so enzymes come along and methylate the cytosines in certain genes, those genes are less likely to be transcribed. So it's kind of permanently or semi-permanently switching off genes. Another thing that can happen, I think less permanently, I guess, is here's the histone protein, and it can have acetyl groups added or removed. So here's an acetyl group, and here's without an acetyl group. So this is, how do we write this? Okay, so histone acetylation. So if acetyl groups added to the histones, then DNA is unpacked or unwound. And if the acetyl groups are removed, then the DNA is packed more tightly. So again, this works by controlling how tightly or loosely the DNA is packaged. And there's a really, really fascinating animation that you should watch if you're interested in this idea. I'll just pop the link in the description below, and here's an image of the video that you should watch. Okay, so the next way that gene expression can be controlled is post-transcriptional. So post-transcriptional means after transcription. So post-transcriptional. Post-transcriptional sort of processing, I guess. And we can kind of, the main thing that this is about introns, exons, and RNA splicing. These are the key terms. Introns, exons, RNA splicing. So what are these things? Okay, so introns, these are non-coding parts of the DNA. Generally speaking, they are non-coding parts of the DNA, which means they do not code for amino acids that end up in the protein. Exons, these bits, are coding. So they do code for amino acids that end up in the protein. So coding, and they will sort of end up in the protein. And the splicing is editing out or cutting out the introns. So here's a picture to show that, and I'll show you an animation in a minute. Well, actually, let's look at the animation first. Okay, I'll turn the volume all the way down so I can just talk you through it. So here we have transcription, and transcription creates messenger RNA. And now we have messenger RNA, that's the yellow and the green stuff, floating around in the nucleus. Now, this green part represents an intron. So this is part of the primary mRNA, that's what it's called, primary mRNA, that needs to be cut out. So we have this complex of proteins. You don't need to know any of the names of these different bits of the proteins, but together they're called the spliceosome. And they all work together, and they kind of bring the two ends of the green segment close to each other. And when they're ready, they snip it off, and they join the two yellow bits together. So they literally edit out that intron region and now we just have the two exons stitched together, and that, I don't know actually what happens to that intron region. Various things can happen to it. It might just be digested, or some other things might happen to it later on, but you don't need to know about that. So that is the process. Okay, so let's label just a little bit more of this diagram over here. So this, the genes in the nucleus are transcribed. Now I'm going to write on here, I'm not sure if you'll be able to see it very well, but transcription happens. And that produces this here, this top band is called the primary mRNA. So primary mRNA is just after transcription. It hasn't been processed yet. And this process of removing the introns produces a mature mRNA, or the mRNA. A few things that we don't really go into too much. It has some sort of extra bit added on one end and an extra bit added on the other end, but you don't need to know that much. And basically the exons are sort of edited together. And this all happens via, where am I going to label this? So to get from the primary RNA to the mature RNA happens via something called the spliceosome. Spliceosome. Apologies if you can't see that. It's a fun word to say. Say it again, spliceosome. So one of the good things about this kind of way of controlling gene expression is it also enables one gene can code for multiple proteins. So this is a little side note. So one gene can code for multiple proteins. Let's imagine, for example, that you have a primary mRNA with let's say there are 4 different exons. Okay, we'll call them imaginatively A, B, C, and D. 4 different exons, and the lines represent introns to be cut out. Now, depending on the way we splice it, perhaps we could get a protein which is A, B, C, or we could get a protein which has the exons A, B, and D. And you could see that we might be able to generate even more than 2 different proteins. We might be able to get several different versions of a protein just by kind of editing it. This would be a little bit like writing a CV, having a lot of different paragraphs, or like a letter if you're trying to get a job. I've done this recently. And then you kind of just edit different paragraphs to kind of tailor it to different places that you want to apply for a job for. It's a good idea if you ever have to do that. And this is what happens. So we'll label that here. It's called differential splicing. And that is my daughter. Hang on a second. So up till now, those 3 ways that we've looked at have all been before translation of the protein. So they've all been to do with transcription or the messenger RNA or the DNA itself. So now we're talking about post-translational methods of kind of controlling gene expression. So some of these things aren't really on the syllabus at all, but just in case you're interested, this is called RNA interference. And basically there are these short interfering RNAs. If you want to know about this, they can bind to messenger RNA and lead to its breakdown. And if messenger RNA doesn't hang around for very long, or if it's broken down quickly, then not much of that messenger RNA that messenger RNA won't be translated very much. So not much protein will be made from each molecule of messenger RNA. So RNA interference is a technique that we can kind of use to interfere with protein synthesis. Like if you want to kind of engineer a cell so that it doesn't produce a protein, you can engineer in some small interfering RNAs to kind of silence a gene. So it can be, sometimes it's called gene silencing. Off syllabus, but maybe worth knowing. This next bit here is about control of translation. And again, this is not really on syllabus, but anything that increases the stability of a messenger RNA will increase the amount of protein produced from it. And anything that sort of increases the number of ribosomes will just generally increase the amount of proteins a cell makes. So we can control the expression of genes partially through modifying translation by manipulating the stability of messenger RNA or manipulating the number of ribosomes in the cell. And if you can imagine if the messenger RNA was somehow made less stable, that would affect it, or if there were fewer ribosomes, then that would affect it. And then the final thing down here, so I'll just put a little star here for this is off syllabus, and this is kind of off syllabus. This one is on syllabus, and it is post-translational. Modification or regulation. And this is pretty simple, really. This is when we have an inactive This is pretty simple, really. This is when we have an inactive form of the protein is made. Okay, so as the protein is made, let's say it's an enzyme, it doesn't do anything. It's not finally ready to get working. So we can kind of have a protein ready to go and only kind of allow it to work in response to a signal. So activation of a protein could happen in a few different ways. So I'll highlight that. Activation of the protein. The most common way is phosphorylation. So if you add a phosphate group, and that's often by something called a kinase enzyme. Kinase enzymes are often ones that add phosphates. So if a kinase enzyme might add a phosphate group to switch on a protein, and e.g. there is an enzyme that you do need to know about. It's in the kind of hormones, hormone communication module. It's called adenyl cyclase. Adenyl cyclase is the enzyme that makes cyclic AMP, and it is activated through phosphorylation by a G protein. And another thing which can happen, it's less on the syllabus, but I have seen it come up in questions before, is some enzymes can have a bit removed. So what will we call that? Partial, I guess I'm going to call it partial digestion of a protein. So like a protein is produced that is too big and then a little piece is clipped off to make the protein functional. So examples of this are trypsinogen is an inactive form of the enzyme trypsin, and it's then converted into trypsin. And actually, there's a protease enzyme that works, I think, in the stomach, that kind of in the small intestine, that cleaves off part of the protein and turns it into its active form. Just in terms of being aware of like biological names, if you see something with inogen at the end, quite often it's an inactive form of something. Another example of this is fibrinogen. Fibrinogen is an inactive form of the protein fibrin, and when that extra bit of it is cut off, it produces fibrin, and fibrin forms clots in like blood clots if there's injury, for example. So let's zoom out. So we've looked at lots of different ways that gene expression can be controlled. And there are one or two little extra bits in this unit that kind of build on top of this. The first is an example of gene expression control through transcription factors. So I'm going to have to draw a sort of link here. So transcription factors, this highlighted word in yellow, I'm going to link all the way around down to here. Okay, Hox genes. Hox genes are genes that control the development of body plans. The Hox genes are transcription factors. And they control development of body plan. Control development of body plans. So how are these Hox genes discovered? Well, they were first actually discovered in fruit flies. And scientists were looking at genes in fruit flies, and they found something called a homeobox sequence. So Hox genes contain this homeobox sequence. So what is this homeobox sequence? It's shown here. So the homeobox sequence, where should I put this? I'm going to pinch in a little bit more and put it here. The homeobox sequence is a sequence that crops up in the genome of this fruit fly here, Drosophila, many times. And it consists of a 180 base pair stretch of DNA. And because there's 180 base pairs in the DNA, this codes for a 60 amino acid protein. Okay, remember 3 base pairs per amino acid. And here it is, okay, this purple bit, let's actually sort of identify it there, is the purple bit, okay? Now this purple bit here forms a certain shape that all these homeobox sequence containing genes have. And it is a helix turn helix, that's HTH kind of shape. It's an alpha helix, a turn, and another alpha helix. You can just about see it here. There's one alpha helix, and there's a turn, and then there's another alpha helix. And it's a DNA binding domain, okay? DNA binding domain. So this is the part of the Hox gene transcription factor that actually binds DNA. So this is a kind of a tricky definition here. Not all homeobox sequence genes are Hox genes, but every Hox gene has a homeobox sequence. Does that make sense? It's almost like a little maths, like Venn diagram sort of thing, like sets. Like if you had a lot like this, you don't have to draw this if you don't want to. This little rectangle here represents all the homeobox sequence genes, and there's like a subset within it that is the Hox genes. Let's do that in blue. Okay, so the Hox genes is a subset of the homeobox kind of containing genes. Does that make sense? So what else do you need to know about them? Well, they are extremely well conserved. So the scientists, when they were discovering Hox genes, as I said, they first found these homeobox containing genes in fruit flies, then they found them in frogs, and then they found them in all sorts of other animals and mammals. So this led scientists to kind of hypothesize and then prove that they are highly conserved. So these Hox genes, talking about Hox genes now, they are highly conserved. What does that mean, conserved? Well, that means that they are present in so many different organisms. Okay, we found out that they actually kind of evolved very, very early on in kind of animal evolution in a type of animal called cnidarian, which is stuff like jellyfish and anemones and stuff like that. So they're basically in all animals, pretty much, because they evolved super early on as animals were evolving from the very first animals. And they occur in clusters. And they're kind of arranged along a chromosome. So here's one cluster, fly, one cluster. But at some point during the development of organisms and the evolution of animals, these clusters got duplicated. So vertebrates, like us, humans, have 4 clusters. So what we think happened was an entire homobox gene cluster got like accidentally copied. It's an example of a chromosome mutation where a whole piece of a chromosome was accidentally copied and it became beneficial because humans have 4 clusters, which potentially gives us an ability to build more complicated body plans, build more complex body plans. Now, 2 other words that sometimes come up, and I haven't seen many questions on this to be honest, but these are things that could come up. There is a word called collinearity, which kind of expresses how they work. It basically means that the sequence of the Hox genes on a chromosome, let's pinch in a bit more on this fly here. Okay, so the sequence of Hox genes on this chromosome is in the same order as which body part the gene controls. Okay, so like the red gene here, this first Hox gene, controls the development of the fly's butt, and this green bit controls the development of this kind of abdomen here, and this blue bit controls the development of the thorax and wings here, make that blue, and so on. So the sequence of the genes on the chromosome is often directly linked to the sequence of body parts in the organism. And what's weird about this is basically if you go in and like edit the genome and like move genes around, because that's what scientists often do when they're looking at fruit flies, and what they basically did was, like, for example, they'd move this Hox gene and copy it and put it here, like at this end, and then what do you get? We get a fly with 2 butts, okay, like a butt at the front, a butt at the end, and the fly doesn't do very well after a while. But like it's that amazing. Like if you change the sequence of these 1 genes, you change the body plan. So what does that tell us? It tells us that these are master control genes. Let's write this down here. Master control genes. So how do they actually do this? Well, how do they control so much stuff? Well, they turn on or off other genes, and they do this via, well, they can kind of regulate the cell cycle to make cells divide more quickly or more slowly, to control the rate of growth. They can control apoptosis, which we're going to look at in a second. So they can cause programmed cell death, and they control gene expression. Okay, so that's pretty much all you need to know about Hox genes. One other thing before we move on, actually, just to do with this conserved idea, because they're so highly conserved, another amazing thing about them is if you take a fly Hox gene and edit it out of the genome and then put it in a frog, it often will work, basically. So if you put a gene that, this gene again, this red gene here, you could do the same thing and take this red gene, more or less, and put it in a frog, and then you get a frog with 2 butts. Okay, I don't know why I picked the example butts, but it wouldn't be a frog with 1 frog butt and 1 fly butt. It'd be a frog with 2 frog butts. Okay, frog butt at the back and one at the end, and the front end, because they are control genes. They control the development of body plan. The gene doesn't code for the entire structure of the fly's behind, but it just says, build a behind here, please. Okay, so that's another thing to do with conservation. Conserved. Okay, so we're almost done. So just last little bit here to do is on apoptosis, which is another way that we can build a body. So we talked about Hox genes, the fact that they can control the development of a body plan, and one of the ways the Hox genes work is through the regulation of apoptosis. So apoptosis we're going to talk about over here in the bottom left-hand corner. So apoptosis is a form of programmed cell death. So cell death that is wanted by the body. So the body doesn't want all cells to live forever. Sometimes cells need to kill themselves, basically to make room. So, for example, one application of that is in the hand, the sort of the building of a hand in the womb. So, if we look over here, we can see, I think this is a mouse embryo, and looking at how the hand develops over time. First, the kind of cells kind of sculpt almost like a mitten, okay, with no gaps between the fingers. And then once all the bones are formed, the skin cells or the tissue in between the fingers is programmed to die, leaving each finger being able to kind of move independently of each other. So the cells in between the fingers need to die in order for the hand to be built. So this is an example of where apoptosis is needed. So I'll just put that over here, e.g. hand. But there are lots of other places where apoptosis needs to happen. For example, when I was at university, in my 4th year, I did research into programmed cell death in the brain. And I was looking at all the signals that went on in these brain cells to cause them to die. And basically this was for the correct development of the brain. So apoptosis is programmed cell death, e.g. development, I'll put over here. But it's also worth noting that also can be triggered. Okay, so apoptosis can be triggered in response to lots of things. So it may not be programmed. It might be triggered by signals inside the body. For example, so let's call it intracellular signals. So if something's going wrong, really bad inside the cell will be like, we're done for, let's just kill ourselves. So that could be DNA damage. So if the cell, this is very good, because if you have cells with lots of DNA damage, they could become cancerous and then cause the whole body to suffer or die. So it's much better that a cell which detects its own damage kills itself than kind of going on to harm the body. So that's intracellular signals, DNA damage. It could also be signals from outside. So the main one for that is a T killer cell. So T killer cells, which you need to know about for your sort of immunity kind of module, they basically will detect if a cell is infected with a virus, and they go ahead and dock onto the cell that's infected, and they use this thing called it's called the death ligand, and it basically just signals to the cell, hey, you're infected with the virus, please kill yourself, and whilst you're at it, destroy everything inside of you. So it's kind of like a self-destruct sequence, if you like. Okay, so that's why you would want to have cells that kill themselves. It seems strange, but how does it actually work? And why is it so good? Well, the reason it's good is because it's kind of neat and tidy, basically. So let's write that down here. It's kind of neat and tidy. And there is another way of cell death, which you don't actually need to know about for the syllabus, but cells can also die not in a programmed way. They can just die sort of accidentally, and that's called necrosis. And necrosis is basically messy and can lead to more damage. So apoptosis is neat and tidy, whereas necrosis is not. So let's label this diagram basically, and let's sort of see it. So here we have the nucleus. And apoptosis here, this is sort of time here during apoptosis. So let's say this cell is infected with the virus, doesn't know it. A T killer cell realizes it and then signals to the cell time to go through apoptosis. So the first thing that happens is the cell kind of changes shape. So the cytoskeleton, zoom in a bit more. Cytoskeleton, breaks down. And that's kind of why the cell becomes a bit rounder, because the cytoskeleton actually defines the kind of borders of the cell. So if the cytoskeleton starts to kind of digest itself, the cell kind of becomes more spherical. What also happens is the chromatin in the nucleus starts to condense. So that's here. I'll just label it down here. Chromatin condenses. And the cell membrane starts to kind of be a bit sort of folded in on itself. We call these folding in bits blebs. And as this progresses, the cytoskeleton breaks down, chromatin condenses, these blebs form. The cell eventually blebs so much that it breaks into vesicles. So into vesicles. And the nucleus itself also breaks into fragments. And finally, these fragments can also be, once they've kind of become small enough, they can be hoovered up by phagocytes. So this is a phagocyte here, which remember is an immune cell, and it kind of just, it's almost like a hoover inside the body. It hoovers up any little pieces of this cell that has killed itself and cleans and tidies everything up. So this is the one vesicle. Okay, let's look at a quick video of this. So here's a little video, and each frame of this video is about 12 minutes, and these are cells from, I think, from an insect that have been sort of caused to undergo apoptosis. So they start off fairly round, but they start to shrink, and their kind of borders get a bit fuzzy because they're kind of blebbing. You can see some of the cells on the left there have completely blebbed and disintegrated into little pieces, and eventually all the cells just kind of almost disintegrate into these tiny little blebs or vesicles that would then get hoovered up. If this was on a microscope slide, if this was in a real living organism, these blebs would be sort of hoovered up by cells in the immune system. So one more thing which is kind of not on the syllabus, but I think just worth knowing, is that throughout this process there are various enzymes that are active, and the major enzyme family that is active is called the caspase family. And these caspase enzymes basically digest the cell from the inside. So not only is the cell neatly packaged up into little pieces that can be digested by phagocytes, but it also digests itself. There are these like self-destruct enzymes, if you like, caspase enzymes that digest everything. And other stuff not really on the syllabus, but also kind of interesting. The mitochondria has a role in kind of deciding whether the cell should in fact go through apoptosis or not. So that's kind of one of the places in the cell that is responsible for regulating the initiation of apoptosis. And once the cell has kind of decided, let's do it, let's go through apoptosis, then one caspase enzyme triggers another caspase enzyme, and we call it a caspase cascade that ultimately leads to apoptosis. Okay, quick few things on that before we completely leave it and we're done. So we talked about development and the fact that it can be triggered. We should also know that in development, there's a balance needed between apoptosis and mitosis. Apoptosis kills cells, mitosis creates cells. So if we have too much, it can lead to cell loss and degeneration. So for example, e.g. Alzheimer's in the brain is caused by apoptosis of brain cells, and this is the thing that I was researching at university, or too little can lead to tumor formation. If cells that should be sort of killing themselves aren't, then you could get tumor formation and cancer. So a lot of work in cancer is working on making sure that cells do go through apoptosis when they should do. Because if a cell has got DNA damage, is potentially cancerous, sometimes the cell's so messed up and damaged that its apoptosis mechanisms aren't working correctly. So we might seek to use medication to restart the natural apoptosis process within tumors so that they can kind of kill themselves And that's one of the areas of research in cancer therapy that's going on at the moment. Okay, so that's everything. We've looked at gene mutations, point mutations, and a little bit on chromosome mutations. We looked at the structure of DNA. We looked at the ways that genes can control their expression through DNA packaging, control of transcription, post-transcriptional splicing, and also post-translational modification. We also looked at a specific case study called the Lac Operon, which is how E. coli switches on and switches off the expression of two genes involved in lactose use. We looked at Hox genes and the way they control body plans. And finally, we looked at apoptosis and why this programmed cell death is necessary and how it's regulated and how it is carried out. So I hope that was really useful. And remember, this was for Module 6.1 of the OCR Biology syllabus. Thanks very much, and I'll see you next time.